Hi, this chapter is dealing with genetic analysis of single population. We are going to talk about what is a population, what is genetic diversity, and how do we quantify such genetic diversity. And we will also talk about what is influencing the genetic diversity in a single population. A definition of a population might be, as it says on the slide, a group of potentially interbreeding individuals of same species and which live within a restricted geographical area. That's a definition of a population, but as we understand, there are always some complication about such definition because there are also some thing that doesn't always fit exactly into a definition. For example, birds, migrating birds, which breed in some northern hemisphere locations, and in the winter they migrate to the south and warmer areas and live there. So there is not a restricted geographic area. Another example of complications to this definition is, for instance, diapause, where we have individuals or, for instance, a seed from a plant, which may stay dormant in the ground for a very long time before it starts growing. Such things might complicate when we are going to estimate the size of a population. Genetic diversity of a population refers to the number of different alleles found in all individuals in this population. Alleles, you remember, is variants of a gene or a genetic marker. And genetic diversity is a very important attribute of a population. When we are studying a population, we are very interested in knowing something about the diversity of this population, and that will also tell us a little bit about how robust the population is to future development of the environment where this population is staying. For instance, when the environment is changing, the genetic variation in the population is necessary for the population to adapt or evolve or adapt to this new environment. So we understand that genetic variation is necessary for the population to evolve, to have evolution. No variation, and we will not get any evolution and adaptation to new environment. And if there is a high genetic variation in a population, the population is much more robust and much more able to adapt to various changes. There are many estimates or many possible methods to estimate genetic variation of a population. Most of these methods are based on allele frequencies and genotype frequencies. In this lecture, we will look into various methods of how these allele frequencies and genotype frequencies are calculated. This is necessary when we are going to also to study a population and see if it is what we call in Hardy-Weinberg equilibrium, as I'm sure many of you are familiar with before. This slide shows us how we can calculate allele frequencies. Allele frequencies we will use later when we also go to calculate the expected genotype frequencies, which is necessary when we are going to test for Hardy-Weinberg equilibrium. This is an example where we are looking into a fish species which might have different color in their skin, and what is deciding this skin color is their genotype, a specific gene called B. If it is homozygous capital B, it will have blue skin. If it is heterozygous, it will have purple color, and if it has homozygous small b, it will have a red color. And we're looking into a population of 100 fish. 28 of these are blue, 34 are purple, and 38 are red. And we will see here, because we know that if they are blue, they should be homozygous capital B. So the frequency of homozygous capital B is 28 fish divided by the total number, which is 100, and then the frequency is 0.28. The same is the observed frequency of genotype. The heterozygous genotype is 0.34, and the observed frequency of the homozygous small b is 0.38. Out of these observations, we can also calculate the allele frequency. The allele frequency is number of the allele we are looking at and divided by the total number of alleles. Okay, so if we want to calculate the frequency of the allele capital B, well, we just have to count how many capital B's do we have. Well, we know we have 28 individuals which are homozygous. Each homozygous individual has 2 capital B's. So we multiply it with 2. 2 multiplied by 28, the number of homozygous. And we add also the heterozygous, 34 individuals, which has only 1 capital B. Heterozygous. So 28 times 2 plus 34, and we divide it by the total number of allele, which is 200. We have 100 individuals, and each individual has 2 alleles. So 200, and that is 0.45. That is the frequency of allele large B, capital B. The same method we apply for frequency of small allele, the allele small b, which will be 0.55. Okay, when we're talking about these frequencies, we often give the code P for the frequency of the dominant allele, in this case capital B, and we have used the code Q for allele frequency of the recessive allele B. And in this case, there are only 2 alleles, and then we will know that P plus Q, the frequency of the one allele plus the frequency of the other allele, will sum up to 1. So this is the method of how we calculate the allele frequencies. So we just saw how we can calculate the allele frequency. If individuals in a population reproduce in a manner that fulfills certain criteria, as we will talk about in the next slide, the frequency of the 3 genotypes that is possible, homozygous, heterozygous, and homozygous for the recessive allele, can be calculated based on the allele frequencies. So if we are able to calculate the allele frequency, we can calculate the expected genotype frequencies. If the individuals in the population is breeding or is reproducing in a specific manner. And if they are reproducing in this manner, and that the expected frequency based on the allele frequencies are equal to the observed genotype frequencies, then we will conclude that the population is in Hardy-Weinberg equilibrium. For a population to be in what we call Hardy-Weinberg equilibrium, there are some criteria it has to follow. We see there should be random mating within the population. There should be no selection of any genotype. All genotypes should have the same possibility to survive. There should be no migration, no import or no export of individuals from the population, and the size of the population should be huge or infinite. It should be extremely huge, and the alleles of a gene we are studying should be inherited in what we call Mendelian, after Mendelian laws. Right. Here we will look at an example of tiger moths, where we see that they have different number of white spots at their wings. If they are homozygous, capital A, they have a high number of white spots. Are they heterozygous, they will have an intermediate number of white spots. And if they are homozygous, small a, they have a low number of white spots on their wings. So if the individuals reproduce in this Hardy-Weinberg equilibrium criteria, then the frequency of these three genotypes can be calculated based on the frequency of the two alleles. And how we calculated the allele frequency we saw on one of the former slide. So the expected frequency, or we can calculate the expected frequency of the genotype, for instance genotype homozygous capital A, that is the probability that the individual inherits the capital A from the father, multiplied with the probability that the individual is or are getting the capital A also from the mother, which will be P times P, T multiplied with P, which is the same as P square. And the same way we are looking at how the frequency of the homozygous small a and small a, that is the probability that it gets small a from the father, multiplied with the probability that it gets the small a from the mother. That is Q square. And what is the frequency of heterozygous? Well, the probability that it get the small a from the mother, multiplied with the probability that it got the large A from the father. But it's also possible that it can be the other way around. It could be the large A from the capital A from the mother and the small a from the father. So that means that we have to take P times Q times 2. Because it's two ways of an individual to be heterozygous. And the frequencies should always sum up to 1. So P square, which is the probability of being homozygous capital A, plus 2PQ, which is the probability for an individual to be heterozygous, plus Q square, which is the probability for being homozygous small a, will be 1. We will use these, what we just talked about, to look at an actual example, as a real example. This is what you see, the box 5.2 in the textbook. You can find it there. I just put out some of the numbers here. As the box says, we will find that observed number of capital A, homozygous with a large number of white spots is 1469. And we have also observed that the number of heterozygous, intermediate number of spots, is 138. And observed number of the other homozygous is 5. And then we wonder, is this population in Hardy-Weinberg equilibrium or not? We can test. Okay, how do we do that? Well, we will find the allele frequencies first. That is the first. Find the allele frequency. And we do that by counting capital A alleles. That is 1469. That is the number of individuals times 2, because there are 2 capital A's in this. Just a moment. Put on this. There are 2 individuals with. There are 2 capital A alleles in these individuals because it's homozygous, and there is 138 heterozygous having one of the capital A alleles. And this will make the number 3076 capital A's in this population. And we do the same for counting the number of small a's, which is 148 then. And then we can calculate the frequency of the capital A and the small a. We see that the frequency is the total number of alleles, 3076 we found here, and we divide it by the total number of alleles, which is this plus this. And the frequency of the capital A is then 0.954. And the frequency of the small a is 0.046. That is the 2 allele frequencies in the population. And then, if the population was in Hardy-Weinberg equilibrium, then we will know that what will be the genotype frequency. Genotype frequency of the homozygote capital A will be p square, as we saw in last slide, and that is 0.954 square, which is 0.910. And what is the expected frequency of the heterozygous? Well, we put it into the formula, which is 2pq. 2 times p times q is 0.0878. And at the last expected frequency of homozygous small a, which is q square, and that is 0.00212. Okay, these numbers are the frequencies of the genotype. So the frequency of the genotype. So then we can calculate the expected number of homozygous and heterozygous individuals. Well, we have the frequency, we have the total number of individuals, which is this plus this plus this, which is the number of individuals. So 0.910 times 1612 is the total number. We get 1467. And the expected number of heterozygous is 142, and the expected number of homozygous small a will be 3. So these big numbers are the expected numbers, and these numbers are the observed numbers. We see they are pretty close. They are very similar. But we can test if they actually are, well, but it's not identical. 1469, 1467, 138, 142, and 5 and 3. So they are not exactly the same. So we have to test, are they actually significantly different, or are these small differences just due to chance? We can test that with a chi-square test. I'm not sure if you are familiar with chi-square test from statistical courses, so we will have a very quick I will show that very quickly. Right, the chi-square test is the formula for a chi-square test is shown here, that we will take the observed value and the expected value, and see the observed minus the expected, and square that and divide it with the expected number. And we sum that for all the genotypes that we are studying. So we see that in this line that we have 1469, which is the observed number of homozygous, capital A, minus the expected number of homozygous, capital A, and square that and divide it by the expected number of homozygous. So that is the homozygous capitals. And then we look at the heterozygous, which is the observed minus the expected, squared, and divide it with the expected number of heterozygous. And the same with the homozygous small a. And when we sum up these three part of the equation, we get these three numbers, and the sum is 1.44. 1.44 is the chi-square value. And to see if that means that there is a significant difference from just coincidence, we have to look into a chi-square distribution table. I'm sure you have looked into such tables before, and there are also computer programs that can give it the exact p-values. But we look at the degrees of freedom, which is 1 in this case of in this kind of type of calculations, because there is 1 degree of freedom in such a calculation, because it's only 2 alleles we are looking at, and if we know the one of the allele frequencies, we also know the other allele frequency, because there should be 1 if we add them together. Okay, so we have 1.44, and we see here, well, it's larger than 0.102. It's also larger than 0.455. It's larger than 1.32 because we have 1.44, but it's smaller than 2.75. So it means it's in between here somewhere. That means that in the text here it says that actually the actual p-value is 0.2. And that means that the difference that we saw in observed frequency and expected frequency are a case of coincidence. They are not significantly different. So in that case, we will conclude that the population is in Hardy-Weinberg equilibrium, because we often put a limit for if it is a coincidence or if there is some specific reason for the different numbers we find at 0.05. This is the often used p-value for if it's significantly different figures. And we see here 3.84. So if we get a p-value which is larger than 3.84 with a degree of freedom of 1, then the data differs so much that we say that it's less than 5% probability that these differences that we observe is because of a coincidence. So since there's so small probability that this is similar, we will say that the population is not in Hardy-Weinberg equilibrium. So we will say that if you get a chi-square value higher than 384, we will say, well, it's not in Hardy-Weinberg equilibrium. Well, we are talking about genetic diversity in populations, and then it is important to have some definitions or some methods or how we can estimate this genetic diversity. We see here a couple of ways of describing diversity. One possibility is use this A, which is we meaning allelic diversity. Allelic diversity is actually just an average number of allele per locus. So if we are looking at one gene or one locus, we will record how many alleles do we find of this gene in the population. And then we are looking at another gene or another locus, and we count the number of alleles in that locus as well, and we find the average number of this. So if we find 5 alleles in one, or 4 alleles in one locus and 6 alleles in another locus, then we have 10 alleles. And if you have 2 genes, then the A value will be 5. So we don't say anything about which alleles are more common than others. We're just looking at the number of alleles. Of course, we understand that this method, the A allelic diversity method, is very sensitive for sample size. Because if we are just collecting a few samples, we might miss some alleles. So if we count more samples, we will more likely find more alleles and also have a higher value of this A. To compensate for that, for instance, if you are comparing 2 populations, and one population is, you have calculated the A value by counting just a few individuals, and the other population you are going to study, there you have collected a much higher number of samples, then there is difficulties in comparing these allelic diversity between 2 populations with different size of the sample. Then you can use this method, which is called allelic richness, and we will not go into the details, but you can look at it in the book. But there we can compensate the population with a fewer number of, a lower sample size with a compensation. We can compensate it. So it's better to compare. But what is in common here is that it is allele size, sample size dependent. A different way of looking at the genetic diversity is what we call the observed heterozygosity. Observed heterozygosity is actually just we count the number of heterozygotes that we find and divide it by the total number of individuals. That will be then you will get a number which is describing the observed heterozygosity. How many individuals out of the total are heterozygous? And a third way of looking at this is to say what we call the expected heterozygosity. How many individuals do we expect to be heterozygous? Well, we touched that a little bit in a previous slide when we talked about the 2 times P times Q, because that was the expected heterozygous frequency when there is only 2 alleles of a marker of a gene. In this equation is showing how we can calculate the expected heterozygosity even if there are several alleles. And what we are doing here is actually just that we are saying the expected number of heterozygosity or the expected heterozygosity frequency is 1 minus the expected homozygous frequency. You see alleles from 1 to m, and then we take the frequency of a given allele and square it, and we do that for all the alleles and sum that, and then we take 1 minus this sum. That will be the expected heterozygosity. Well, here you see we call it a small h and call it gene diversity. That is often the same as expected heterozygosity. Especially, well, in most cases, we treat them as the same thing. In a population that is in Hardy-Weinberg equilibrium, we will find that HO, that the observed heterozygosity, is similar or the same as the expected heterozygosity. But sometimes we find that the observed heterozygosity is smaller or less than the expected heterozygosity. And what may be the reason for this? So what could be the possibility of what could be the possible reason for that? Well, one reason could be what we call the null alleles. What is a null allele? We talked about alleles, different versions of the gene or of a marker, but null alleles is something that we call them. If you're using PCR for amplifying your samples and amplifying your gene or your gene marker to see if it is present or not, you will find, for instance, if you're running PCR, you'll have two bands. One is the one allele and the other band is the other allele. And if it is a homozygous individual, you will have just one band. If you are an heterozygous individual, you will have two bands. And if you are homozygous for the other allele, you will have one band at that position. So if you see two bands, you will see heterozygous. And if you see one band, you will see homozygous. Of course, we know there are some alleles that are not possible to see, to discriminate between just a band, large and small bands at the gel. But for instance, if the case was that. But if we see just one band, then we will say, well, this is homozygous. But it could be that the individuals actually was heterozygous, but that the PCR primers that will amplify this allele doesn't or didn't fit, because there had been some mutation in the area where the primers should hybridize, so it doesn't work. So only the one of the two alleles will be amplified, and we will record this individual as a homozygous. So that is, we think that there's a lot of homozygous individuals here, but in real, it is several of these were heterozygous. So we count more homozygous than heterozygous. So the observed heterozygous will be less than it actually should be. So one possibility is these null alleles giving the wrong impression of a lot of homozygous, and because of this problem of detecting null alleles. But when we have ruled out this and also other possibilities for finding these differences, you can see it in the textbook. Then if we still see there is a difference between observed and expected heterozygous, one reason is that, well, maybe the population is not in Hardy-Weinberg equilibrium. And the reason for not being in Hardy-Weinberg equilibrium is these criteria that we saw in one of the first slides. Not random mating, not, well, a very small population, there is migration, and there is some natural selection going on, and things like that. Well, I'm sure that most of you or several of you are familiar with several of these things we are talking about now, because most of you have some ecology or population genetics courses before. But we still touch these things because they are so central in a lot of these molecular ecology studies as well. A central factor influencing genetic diversity is genetic drift. Genetic drift, as you know, that means that the allele frequency in a population will change from one generation to another, sometimes very little, but also sometimes very large differences from one generation to the other, just because of chance. Just because of random mating, or well, randomly which individual will be made with another, the allele frequencies will differ from one generation to the other. And that means that the genetic drift will lead to a fixation of one of the alleles at a locus or at a gene. Because just by randomness or by chance, one allele becomes a little bit more common than the other, and then it will be a little bit more common. So after some generations, it will either go extinct, that means it will disappear from the population, or it will be totally dominating, having the frequency of 1. There is several things influencing this genetic drift. In a very small population, it is very most notable, this genetic drift. Genetic variation is lost much faster in a small population than in a large population. And genetic drift is also closely linked to also another very important measure of population genetic structure, what we call the effective population size, N or NE, which is the effective population size. This is a slide which was not in the original slide or PowerPoint I put out in Canvas, so maybe not all of you have this, but it's just an extra slide showing a computer simulation, showing that in a small population the genetic drift is much more easy to see. Here we see in 3 populations with a size of 20, 200 and 2000. And there is a computer has simulated an allele starting with a frequency of 0.5. When it after the generation goes, in some simulation it will stay like this, and then suddenly it grows and it goes down, so it goes up and down. In this case, it goes right up just by coincidence, and then it is the frequency of 1 is fixated. And in other cases, it goes smaller and smaller, and it ends up with being extinct. Some of them, one allele can survive for 50 generations, but it's absolutely much lower than the starting point, which was 0.5. So sooner or later, it will go down to extinction or fixation. The same happens here when the population is larger, 200, but it goes slower. But some are slowly growing and going more and more close to fixation. And here in this case, we have a much larger population, 2000. Nothing much has happened after 50 generations, so it will take much more time, much longer time before any fixation will happen. So we talked about population size, and we talked about in the last picture, last slide, that a small population size was much more vulnerable for allele fixation or allele extinction. So we have different ways of looking at population size. Here you see two ways of looking at population size. We are both talking about the census population size, NC, and one definition, there are several, but one definition is just the number of individuals that are in a particular population. That's quite easy, we think. It's just to count the number of individuals. We shall see later that it's not always that easy, but well, the number of individuals. But the effective population size is sometimes a little bit more complicated, maybe, but it's more interesting or more accurate, because the NE, or the effective population size, that is something that reflects the rate at which genetic variation is lost because of genetic rift and also inbreeding. So as we saw in the last slide, that we saw if the population size is 200, the drift or the genetic variation is lost very slowly. But if the population size is just 20, the genetic variation is lost much more quickly. So if we think that we have a huge population, but the effective population is maybe not that big, because one reason or the other we shall see, but maybe there's just a few individuals in this population that are actually breeding. Then the effective population is much smaller, and we would expect that the genetic variation will be lost much more quickly. Usually, as it says here, usually the effective population size is much lower than the census population size. So how can we quantify, or how can we quantify the census population size, and how can we quantify the effective population size? You will see in the textbook there's several methods, and there's a lot of reading and text about these things. The important for you is to see that there are a lot of different ways, and the simplest way of finding or quantifying the census population size is just to count the number of individuals in the population. Then to find the census population. But there are some challenges. For instance, you can think about of population consisting of a sample that the species that gets enormous amount of offspring. There's a lot of individuals in this population, but a huge part of this population is just larvae or small offspring, newborns, and just a tiny fraction of this huge number of offspring will end up with as mature or grown-up adults. So in such cases, maybe it's better to look at mature adults as the number of mature adults as the census population size. So it differs. It depends on which species and which population and which conditions that is in. But simple way, count number of individuals. And then how can we quantify the effective population size? Well, it is a lot of ways, and there is no specific rules for this, but well, there are rules, but there are different ways, and sometimes it is necessary to combine different methods as well. And it can be estimated from both demographic data, that means how a population behave and how it is, and you can also be at genetic data, that we can take samples from individuals or from organisms, and we can analyze it, and we can take some conclusions about the effective population size. So there are several methods, and some are based on collecting samples or doing observation in a single generation, while others are based on that we collect samples from time series from several generations. Well, at least there is one generation in between these two sampling, so that we can see, we can compare, and we can draw some conclusion about how many individuals are actually involved here, and how what is the effective population size. Just a short example of NE, how NE, or the effective population size, is very different from the consensus population size when we are looking into demographic data. For instance, these sea lions are examples of huge population, and there's maybe 50% of males in this population, but there is only a few of these males that actually breed. They are competing and make sure that there is only a few males that will breed with a lot of females. That means that the effective population size is very low compared to the consensus population size. This slide shows us that the genetic diversity, and also then the effective population size, is reduced after population bottlenecks and population size reduction. You see in scenario 1 here that you have a population. There is a population bottleneck. It is reduced by some reason, for some reason or the other. So that will mean that the genetic variation is lower, because a lot of individuals here has been well removed. There is just a few. The population has been small. But maybe, well, and then we see that this small population has regained, so it has grown, and in this case it is the same size as it was before, but we still see that the genetic diversity has gone down. In this scenario, you have the same population as here. It has gone through a bottleneck, and it ends up with a smaller population, just like here. But in this case, there is not this situation of regaining, that it grows again, that population will be large again. There is it keeps its small size, and maybe even becomes smaller, and there is genetic drift, and making so that some alleles will go extinct, and some allele, or one allele, will be fixated. So, and this population then, even if it should grow later to become larger, but of course then there will not have any larger genetic variation, or genetic variation, then it will be much more vulnerable for future environmental changes. It doesn't have this variability to respond to a change in environment.